Good morning or good afternoon, everyone. Welcome to Genome Webinars. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Imputation Strategies and Techniques for Analyzing Complex Traits. This webinar is sponsored by Almaden Genomics. Our speakers today are Dr. Pooya Yazdi, Chief Science Officer and Chief Medical Officer at Self-Decode, and Dr. Mark Kunitomi, Chief Scientific Officer and co-founder of Almaden Genomics. You may type in a question at any time during this webinar, and you can do this through the Q&A panel, which appears on the right side of your webinar presentation. We will ask our speakers your questions after the presentations have concluded. Also, if you look at the bottom tray of your window, you will see a series of widgets that can enhance your webinar experience. So with that, let me turn it over to Dr. Yazdi. Please go ahead.
Thank you very much. It's a pleasure for me to give this little presentation virtually to everybody out there. So let's just get started. I'm very excited to speak with everybody about genotype imputation. Now, this is something that's quite popular, all right? So there's a couple things we're going to go over today. First, we're going to just generally talk about the general idea behind genotype imputation, what it actually is. Second, we're going to go through some various models and their strengths and limitations. And finally, we're going to talk about a new imputation method that me and our team have developed at Self-Decode that I'm quite proud of. So we'll talk a little bit about that as well. Okay, so let's just dive right into it. Simply put, genotype imputation is when you predict missing genotypes. Think about it this way. If anyone's had any kind of DNA microarray done before, you'll typically get around 700,000 genotypes, right? So the idea behind imputation is, well, we have that information for those 700,000 genotypes. Can we use that information to figure out other genotypes that you have that were not part of the test? So this is really, really popular in GWAS. If it wasn't for this, GWAS would just simply be not as powerful as it currently is. And it's increasing in popularity for many reasons. One, as we'll discuss, it increases power in GWAS. It allows you to do meta-analysis when you take results from multiple different GWAS and you can combine them to get a boost in power. And finally, it involves fine mapping of causal variance, right? GWAS is called GWAS because it's an associative study. It's, when you find SNPs that are associated with some disease, but, association is not causation, right? So the whole idea behind that is eventually to try to pinpoint down what are the causal variants from the associative one. And that's another thing that imputation is important for, to boost power on that. And simply put, the entire reason why this is even possible and what all methods currently leverage is linkage disequilibrium and recombination. Simply put, like the DNA you inherit comes in chunks from your mom and dad, who inherited in chunks from their mom and dad, and so on back in evolutionary timescales. And as you go further back in evolutionary timescales, you have more and more recombination that breaks up those chunks. Now, this is great. We all learned this in intro biology class, but what it also tells you, though, is that you could use this to find missing information, because it's not like you're getting those genotypes inherited individually. It always comes in these blocks, right? You inherit them in blocks. And that's kind of like the theory behind this. So let's go through an example of how this works and why this is important. Okay? So as you see in the bottom left over here, this is classically what you get. You'll get a microarray done. Let's say you wanted to do microarray sequencing in like 100,000 people, and you have a list of sites on the microarray that the machine gives you the genotypes for those people. Now, if you did GWAS just on this, and you associate with some disease, you might get something like these values for these genotypes. As you can see, there is an association there, but it's also not very clear, because typically a microarray, you get 1 or 2 genotypes in certain chunks, and they're very spaced far apart on the genome. So it's not the signal to strength ratio is not that strong. But what you can do is first phase them, which is when we just put them into mom's chromosome or dad's chromosome, and now you have phased genotypes. And you ask this question, the ones that are missing, the ones that we didn't genotype directly with the machine, can we infer what those are based on some other parameter, right? And that's basically what it allows you to do. Because what you can do in genotype imputation is you have a reference of a bunch of people who had maybe whole genome sequencing data done on them, right? So you have all their genotypes, not just the ones you genotyped in your study, but in this reference population, that you know all the genotypes that they have, right? And based on this, and taking into account the fact that we all inherit DNA in chunks, in these kinds of linkage disequilibrium chunks of DNA, we have built statistical models that use this reference and this information in order to guess what your missing genotypes are. Hence why it's called genotype imputation, right? So, you know, over here you can sort of see it, and we'll get a little bit more into how this is done further along, but it's almost like a way to think about it, and the color coordinates kind of show it over here, is you can start thinking of, okay, I'm inheriting them in chunks, and so we have these chunks. How can we kind of like stick these chunks together in order to create your DNA in this person's DNA, right? And so that's what you almost do over here. It's almost like we're staking these chunks from the reference, saying what looks similar to what we have, based on that, sticking different parts together. And from that, you can infer these missing genotypes. And as you see, then when you perform your GWAS, you suddenly get a big boost in power. And it makes it easier to find that the signal strength is over here, in this one that pops up, right? So let's go through another example. So you'll see this one really clear. So, you know, your black are all the observed SNPs. These are all the single nucleotide polymorphisms you had in your microarray kit. These are all the ones that were sequenced directly for all your test subjects. And then you used imputation, and you got a bunch of other SNPs to go along with it. And as you can see, the signal becomes a lot easier to pop, a lot easier to see, a lot easier to discover once you combine these kinds of imputed SNPs with your directly genotyped SNPs. And you can, and this kind of helps you detect where the causal variants have been, right? So it allows this kind of fine mapping. It allows this boost in power, all these things that we're highly, highly, highly interested in. All right, so how does this work? We'll keep discussing this a little bit. But basically, every single genotype imputation method that's currently used, or that's popular to be used, all basically work with the same statistical framework, which is called a hidden Markov model. Now, this is a very useful but complex model, but it basically works with this assumption. And so we'll talk a little bit what Markov chains are, what hidden Markov models are, but the basic idea is this. You have an observation, which in our case, our observation is the observed genotypes. It's the genotypes you are directly sequencing from the microarray machine that you know exactly what those genotypes are. And the goal from that is to infer some hidden state, which is a reference haplotype, right? So it's a chunk of DNA that you're trying to infer based on only observing the genotypes that come off the machine. And so the idea was, what kind of statistical model can you use for this? And a hidden Markov model works perfectly for this. Now, the reason for that is a hidden Markov model is called hidden because the thing you are trying to decipher is hidden, is not observed directly. And the reason it's called a Markov model is based something we call the Markov property, which is the idea that any event or any state is only influenced by the preceding state and not anything before that. So let's go through that and make it very simple. So in our situation, it would be a very way to think about it is, if you're trying to determine which haplotype somebody belongs to based on just observing their genotypes, the only thing that matters is when you're looking at the current haplotype they're on. So when you're trying to figure out the next haplotype, that is only influenced by the current haplotype they're on. And all the haplotypes that came before that, like all the tons of DNA that came from all the other chromosomes, don't really matter, right? So that's a Markov property because it's only really dependent on this state, right? And so based on this, you know, they have these nice fancy hidden Markov models. They have these great algorithms for that. You can basically run these algorithms and kind of decipher what's going on. Now, I should also tell you, that make it very simple, but the guys who figured out how to do this actually got a very big paper, and it's quite popular, and it's called the Lee-Stevens hidden Markov model after two guys, Lee and Stevens, who came up with it. Population genetics is a really big, long field that has like over 100 years of research in it, if not more. And you know, they've come up with tons of statistical models to explain evolutionary forces, to figure out recombination, to figure out, you know, all this information. But the issue was, most of these models, you know, while they were great mathematically and they made a lot of sense and they were beautiful, you couldn't actually use them in real life when you started getting genotype data because they were computationally intractable. It wasn't something you could easily solve and have a computer infer. And so what happens is Lee and Stevens came up with a model, and they originally used it to figure out the recombination rate. That was the original thing what they used it for. And then everyone else figured out, well, you can use the same model to do other things like genotype imputation. And it's a very simple model. It's a haplotype copying model, and it forms the basis of everything, not everything, but everything in genotype imputation and phasing and a bunch of other things in statistical genetics. So it's a probabilistic generative model which supposes that a sampled chromosome is an imperfect mosaic of other chromosomes found in a population. So let's put this into normal English, right? Which is that if I am trying to find out what your chromosome looks like, you can make an assumption that a way to think about what your chromosome looks like is that if I had the chromosomes of a bunch of other people, your chromosome would basically be a mosaic of all these other people's chromosomes, because, you know, we all had ancestors and we have tons of common ancestors. And if you stuck their pieces of their chromosomes together, you would reproduce your chromosome, minus one addition, which is that it's an imperfect copy. Why? Because there's mutations, right? So we don't always keep the same exact DNA that our ancestors had because you infer mutations. And so this was like a very simple deduction, but it's a very powerful deduction, because once you make this deduction that you can view someone's chromosome as an imperfect mosaic of other chromosomes found in a population, then the idea is like, well, how can we use a statistical model in order to figure this out? And so you can think of it as almost like a copying path through chromosome space. So you have a bunch of people's chromosomes data, ones and zeros, whether they have the reference allele or the alternative allele, and you can view the person's chromosome as literally almost taking a path through the space. Does it go up? Does it go down? And there's a graph. Next slide will show what we mean by that. And this is how you can kind of stick people's chromosomes together and for your person's chromosome. And now there's a couple things you should know. While it's a population genetic model, and you know, there's always these two users specified parameters, theta and rho, and usually they're interpreted as the rates of mutation and recombination. That's what they're thought of. The actual values that go into these algorithms have no bearing on reality, what's going on. Just so, and this is always a point, because the first time I ever encountered this, I was like, this cannot be right. This recombination rate is way too large. This mutation rate is way too small, but it's just the parameters you have to use for the statistical models to work and give you good results. So the biological phenomenon, they actually represent this off. So if you're a biologist and you see that, you go think something wrong. It's just how it works. Okay, so here's what we mean. So we have your study sample here. You can easily see what the genotypes you've observed, the ones that come off the machine. And then you can see all the reference haplotypes. And you can see what we mean here. Now, real quick, before you even do imputation, remember when you get the genotypes off the machine, it always comes as genotypes. It doesn't come mom's chromosome, dad's chromosome split up. So there's always a thing called pre-phasing. You always have to phase the data before you impute the data. Oddly enough, not surprisingly, the same exact algorithms, the Lee-Stevens model, is also used as a phasing algorithm, right? So it's the same idea. You can use it for phase. So let's say we phased our data. We've put a mom's chromosome, dad's chromosome, and now we have our user data, and we have these 3 sites that we know their genotypes for, and we've already phased them. We know which chromosomes they belong to. Let's figure out how do we create this path through this chromosome space? How do we go through these reference haplotypes, stick them together in order to create this person's haplotype? And these hidden Markov models do exactly that. These statistical algorithms, they're already fully developed. You just have to apply the algorithms. And based on that, you kind of stick it together, and it's literally, you'll see the mosaic in this picture. And based on that, now that you've stuck them together, you go, well, it's very simple. So now I know this reference and then this reference, we stick them together here. All those missing question marks where you didn't know the genotypes for, you didn't know what alleles they had, just take them right off your reference, and now you have it. And it's quite powerful because it's quite accurate. Now, let's go to something we did with this, which is, since there's all these different tools, there's all these different parameters, we wanted to figure out, okay, so what's a good imputation tool? What's a good phasing tool? What's a good reference set? Let's test all these out. And so, I'm making it sound very simple. You got your data over here. You have your reference populations over there. You run a simple hidden markup model. You get your results. Well, that's all great, but you also have to get the data in the right format in order to do this. There's a lot of pre-processing that goes on over here. So, for instance, you always are going to get raw data of some sort. It may be data on a different genome build, genome 37 versus genome 38. It may not be normalized and so on. So there's all this pre-processing. And most of this, there's a bunch of tools to do all this pre-processing. So for instance, in our case, we had to take the raw data. We use BCF tools to, prefix the annotations, get the right annotations in place. To split multi-allelic sites. These programs don't work well on multi-allelic sites, so you typically split them. And then you have to normalize, you have to make sure that, all your variants are kind of like in proper order. They're left aligned so that when you say like the variant, the reference is this position, we know exactly what it is. And then the alternative allele is exactly fixed into that position. You have to get rid of sometimes rare variants. What we mean by that is like, singleton, for instance, is a mutation you find in an individual. You can't impute that, right? So if you have in part of your reference files, there's a bunch of singletons in there, you typically remove that. If you're only finding 1 or 2 copies, those are singletons. You're not going to be able to impute that. It just messes up your data anyway. And so on. So you have to do all this like processing to typically get started. And then what we did is we did all this processing. And then we tested out multiple variables, right? So we basically did 3 different phasing algorithms versus 3 different imputation algorithms. Like I said, you'll notice Beagle 5.4 is in both of them. It's both a phasing and imputation algorithm. A lot of them, once again, they're all Lee Stevens hidden markup models, so it makes sense that you would do it for both. And we also tested 2 different reference panels. Both of them are the 1,000 genome project reference panels, but there's different iterations of that. There's the 30X and there's phase 3 and so on. And part of our work over here was, we tested out all these combinations in order to try to figure out, like, is there an optimal combination? So in total, we ended up testing out 36 different combinations to get our results. Now, we're going to show a very, very cool video over here, which I'm quite excited to show. So let me get this started. Okay, so this cool video over here. So part of the thing is we decided to build this on the Almaden Genomics genome platform because it kind of makes testing these things out much more efficient. It saves a lot of time because, as you'll notice, there's tons of different variables you can change on these things for your software. And if you're using like command line tools, you're buying from mathematicians that are going to pull their hair out because they're constantly have to be writing code. So on the genome platform, you can get this stuff running. You can get it operational. It's almost like a plug and play. It's almost like a video game in many ways. That's what I love about it, actually, because it looks fantastic visually, and you can get it in there. And based on that, you can kind of speed this up. So if you're trying to figure out what the best parameters to use and so on, you can get that going and move very forward. So it's quite cool, and it basically ended up saving us a lot of time to play on it. And the other thing that's great about it is if you have a genomic scientist who know how to, don't know tons of code. These are not data engineers. These are not like hardcore bioinformaticians. They can still get something like this operational and still get you the kind of quality data, which always I love hearing because I was a physician and I was never really trained to code properly. So anytime I hear about anything that makes it easier for me to get stuff up and running, that's exactly what I love to hear. So, okay. And as you see, I kind of, stole a little bit of thunder over here. But typically on these kinds of pipelines, one of the real frustrating things in bioinformatics is you always takes forever to get a pipeline going. You always have to iterate over it. You always have to fix stuff. And typically, you'll see stuff like, 2 weeks, 3 weeks, 8 weeks. In total, you're spending like 13 weeks. In total, you're spending weeks to months to build something, right? On the genome platform, because a lot of the utilities are already there and it's simply plug and play, drag and drop, stitch them together, you're going to save tons of time. So in our use case over here, you're basically saving about 13 weeks. 13 weeks of work just to get the pipeline operational to do all these kinds of testings. So, a 70% reduction in runtime and 25x reduction in runtime costs. It's going to save tons of money. And and like I said, For me personally, what I love about it is you can have actual scientists who know a little bit about computers working with these kinds of data sets without the need to have, like, you know, a team full of bioinformaticians running around and writing code. That to me is by far the coolest prospect. That and the fact that, you know, you can run through all these parameters, you can always test for multiple of these things and kind of get this, change things around and iterate really quickly to kind of get at your solution without spending weeks and months of writing code for every step. Okay, so we used that. We got all those testing done. So what did our results find? Now, there's a couple of interesting things. So this is phasing, all right? So once again, remember, phasing is like imputation. Phasing is when you just, you know, split the mom and dad's chromosome, get them on the right one, and then you can do imputation. So phasing affects imputation, right? And so there's basically two different ways you can phase. You can phase reference-based or reference-free. Reference-based is as simple as before. You have a bunch of reference samples in a population, and based on these reference samples in a population, you use that to phase one sample at a time typically. No, it doesn't have to be one sample at a time, but a good typical use case for that is one sample at a time. And, you know, as a direct-to-consumer genetics company, you do that all the time, right? When you get the genotype data, it's not like it's some study where you had 300,000 samples all show up at the same time. So you're getting one at a time, and as you're getting it, you're typically running it through with a reference set for phasing, and based on that, you get your results. And as you can see, it's pretty accurate. And then there's also reference-free. Now, reference-free is, you know, there is no reference for phasing. This is typically used when you have thousands to hundreds of thousands to millions of samples. It's almost typically done in a study, you know, and we phase them all together without reference. Now, first thing you'll really notice is the difference between accuracies for all these, you know, different software, or even, you know, reference is minuscule when you typically think of like accuracy measures, right? 93.4% versus 94.15%, you know, it seems minor, and, you know, most people are going to brush it off. Now, depending on your use case, though, this could matter. Remember, the genome is quite large. You're talking about, you know, billions of pieces of information. So even 1% is, you know, a lot of differences, right? So it really depends on your use case. Now, one of the things I really like to tell people is when you go reference-based or when you go reference-free. Reference-free makes a lot of sense if it's typically a study in which you genotyped a bunch of people on the same exact machines after you collected all the samples. One of the reasons why it works so well when you do reference-free is that the machines always have uncertainty in them. Remember, you know, the rules of physics don't stop just because we're doing biology here at genetics, right? Any kind of machine that measures anything is always going to have a little bit of uncertainty. There's always going to be inherent biases in the machine. There's always going to be runs that, you know, where the signal intensity of the laser was a little off and so on. And all this produces kind of these kind of biases in your data. Now, when you do these kind of reference-free methods, what happens is because they were all done with the machine at the similar time, a similar workflow, it's almost like baked into it, and like, you know, the algorithm can sort of figure this out and still, you know, phase them properly on the two. And you don't have that benefit if you go reference-based. With reference-based, you know, it's one sample at a time. But if you're, you know, a direct-to-consumer genetics company, or you have different kinds of machines using your microarray data at different time points, you know, at first one we did at this location, the second one we did at a completely different location way later, and so on. That makes sense as a reference base, right? You're going to run those through reference base, and based on that, you're going to decide which one to do. You know, maybe in your case, use shape it for, you know, and if you're doing reference free, you know, shape it for also works well. You know, so you have these kinds of choices. Like I said, the difference in accuracies are very, very minor. So you have to really, really think about what your use case and what the question is you're trying to answer. If it's something where, you know, the accuracy really, really matters, and we'll actually get through a case why we've developed our own imputation method, then, you know, you want to make sure you're picking up the most accurate one. If your use case, the accuracy could be, you know, what's 1%, you're just interested in something else, maybe you'll pick on computational efficiency or cost or something like that. So now that was the phasing. Now we'll talk about imputation. Now, one of the things you should know about imputation is, we'll go through some of this information, is imputation is really, really kind of dependent. The quality of your imputation is dependent on multiple factors. So it's not a one-ship-all metric. Total accuracy really is not that meaningful when it comes to imputation. And so in this case, you should know that IQS basically stands for imputation quality score. people in the field came up with a different metric than accuracy in order to gauge imputation accuracy. And the reason they did for that is that chance always plays a factor, especially, you're imputing a variant that's found in 95% of the population. Even the crappiest model is going to start guessing that right after a while, so you kind of want to take all this into account. So they came up with this IQS score as a way of doing it. And here what we did is we basically broke down minor allele frequency stratification of these imputed variants to figure out what happens as your minor allele frequency decreases. Now, the way this figure is set up, the real take-home from the figure is something that's not surprising if you've read other papers, is that as your minor allele frequency decreases, especially when you get to 1% and lower, imputation just falls off a cliff. This is typically the reason why a lot of times people will cap their GWAS at like some common allele frequency. And the reason is, imputing rare variants is very hard. Anything less than 1%, it's typically highly, highly inaccurate. Now, there's ways to get around that. One way to get around that is if you have like a massively deep reference panel. Like for instance, there was a paper published last year in which they started releasing some of the UK Biobank whole genome sequencing data. So they had 150,000 of the 500,000 samples, deep sequenced, and they used that to impute the rest of the UK Biobank, and they were able to impute lower than 1% because of this. But you may not have access to 150,000 samples for your job. And even if you had access to 150,000 whole genome sequencing, that might drive you, it's very expensive to use that reference panel, much more expensive. So you may want to cap yours. But the basic idea is that especially when it comes to these rare variants, anything less than 1%, typically imputation is not very accurate. And as you go higher, it becomes more accurate, so just makes intuitive sense. The more common an allele is in a population, the more times you see it in your reference population, the better your algorithm is at guessing who has this and who doesn't have this. Very straightforward. But here's some more interesting stuff, at least I find it a little bit more interesting, is that it's also affected by the ancestry of your data set, whether it's the ancestry of the actual reference samples you have or the ancestry of the user or the one you're trying to target population. And so ancestry also affects your imputation quality score, which is another reason why you want to look for, if you're imputing tons of variants with a broad ancestry background, you want to have a broad reference sample that has, just not white Europeans in it. You want other kinds of ancestries in there if your users or your targets also have that. This graph basically shows the same thing, is that, in this one, it's a little bit different because in this one, what they did is they go, okay, we're just going to use the same reference, the same 120, 120 samples for everybody. And now we'll just look at what happens to different users with different ancestral backgrounds. And not surprisingly, that, like I said, both whether you have which ancestors you have in your reference sample and the target ancestry also affects imputation. So as you can see, there's some limitations there, which is sort of why we kind of developed our own imputation method. We're not going to get into too much of the technical details. I'll kind of blitz through this a little bit. But basically, we wanted it for a specific use case. Rare variants. We're very interested in rare variants because rare variants have much higher medical significance than common variants are. They typically have much larger effect sizes when it comes to disease and so on and so on. So we're obsessed with this idea of like, we should be able to get much better at imputing rare variants. So we specifically tried to come up with an imputation method that was much better at doing rare variants. Like everything else, it uses the Lee-Stevenson Markov model. But what we did was slightly different, is that we basically did this really cool algorithm called the bidirectional PBWT. PBWT stands for Positional Burroughs-Wheeler Transform. Before you start Googling and then try to figure out, the best way to think about it is, it's just an efficient algorithm for storing, compressing, and searching genetic data. At basically what it is. And the reason it's bidirectional is that it does this in two directions. So you don't just go, left to right, you can also go right to left. And so this is very ideal when it comes to imputation. So if you have a SNP of interest, you have a marker of interest, you can search both in one direction and then the other direction. And based on this, you can find what the potential haplotypes are that are most likely to be found in this target one. And The other thing we did over here, I shouldn't even say really, it's really the, the scientists I had who are working on this, who might be with here, Adriano and Abdullah. Basically, the other cool thing they did is that a lot of times, you and somebody else might share an allele at a specific population. And this could, and this is either shared because it's identical by state or identical by descent. If we know for sure that we share the same allele because we inherited from a common ancestor, we call that identical by descent, as opposed to identical by state. And why does this matter? Because, well, that is what you're looking for when you're doing imputation. What you're really trying to find is, let's find people who share a common ancestor with me and hence would share alleles that are identical by descent. So they also wrote this really cool heuristic for making it easier, being more precise of when you think we have an identical by descent. And they typically ran the same hidden Markov model, and they used the forward-backward algorithm to compute the necessary data and so on and so on. But here's the data. Okay. So it's a very cool algorithm. It's, like I said, it uses Lee Stevens. We just did some minor tricks because we were interested in making it easier and better at, finding where this mosaic is, because that's what it's really about. The Lee Stevens came up with this idea. We want to find which mosaics to stick together. And, what they did is they basically came up with this cool way of sticking these better together. And, we did a little test. We compared it against Beagle 5.4, Impute 5, and Minimac, which you saw previous data for. Now, this graph, to make it easier to visualize, basically we just looked at the errors, and we did like a log fold enrichment of the errors. So it's just counting how many errors each have. So the lower the number, the more accurate it is. And we binned it into different minimum allele frequencies. And as you can see, the green is selfie, which is the model that they came up with. This consistently performs better at all minimum allele frequencies compared to the others. But it's really at the bottom here is why we had done all this work on this. Is it's much better at imputing these rare variants, which is really what we're interested in. And the same thing, we did it. We wanted to make sure, because like I said, the ancestral origin also affects this. We tested this in different ancestries, African, East Asian, European, South Asian, and consistently, once again, the green, which is selfie, it was much better at imputing overall compared to the other ones. And so, with that said, let's try to get a little summary of what we discussed today, and then move forward. Okay, so like I said, imputation has tons of benefits, right? Let's just repeat some of them because this is the important stuff. It increases statistical power by predicting unobserved genetic variations, right? Lee Stevens hidden Markov model. The whole idea is like, whenever I'm looking at anybody's DNA, whether it's my DNA or anybody else's DNA, if you're looking at the entire chromosome that somebody has, we can think of it as an imperfect mosaic of other people's chromosomes, right? So once you have an imperfect mosaic of other people's chromosomes, we can infer what is going on from there based on that, right? And it's cost effective, right? If you want to do whole genome sequencing on, your 150,000 samples, that is going to cost a lot more than taking those 150,000 samples and doing genotype imputation on them, and then followed by a microarray, and then followed by genotype imputation on them. So that's much more cost effective, right? And then it builds meta-analysis. What do we mean by that? There's different GWAS, right? Group A did a GWAS here, and one point in time, Group B did another GWAS later. You can take that information and then do a meta-analysis with imputation in order to combine those results into one meta-analysis, which boosts statistical power. Enables fine mapping. Once again, if you have more information on more of these variants, especially the rare variants as well, that, why we're working on that, you can start fine mapping. It's no longer about associations, it's about causality. Okay, so which variant actually causes this disease, right, is causal in this case. Like we said, imputation accuracy varies across ancestral populations. Our own algorithm, Selfie, achieves higher accuracy in variants across various ancestral populations, and also, as importantly, is much better at rare variants. So those are ones we're the most interested in. Bioinformatic pipeline development for application-specific use cases of imputation requires testing out many tools and parameters. As you saw in that, you got to constantly change out some of these parameters. Each one of these parameters can affect what your use case is. So you have to constantly be testing these things out, which is ideally why something like the Alamedin Genomics genome platform is an ideal use case, because you can start testing those things out, and based on that, get much higher turnaround times with your data. So I will now head this off to Mark and to take the rest of the presentation. Thank you.
Thanks so much, Puya. Yeah, no, it's really fantastic work. I have to say, from the first times I saw the work on the different ancestral populations, it really blew my mind. And of course, seeing the way that Selfie is able to get higher accuracy for imputation, not just within the large corpus of data, but across different ancestral populations. I think that it's just has tremendous potential. So I'd like to tell you guys a little bit more about our platform at Almaden Genomics. I think that from working together with Puya's team, we kind of learned that the key to successful imputation strategies is to employ lots of different toolings, explore lots of different parameters, and then be able to move from the types of proof of concepts that get developed to solutions that kind of scale seamlessly to the large amount of data that they need to be applied to. And this is really where our platform at Almaden Genomics Genome, it really shines. First and foremost, it's kind of an intuitive drag and drop graphical user interface combined with a curated library of tools and pre-built workflows, which allows you to rapidly collaborate, build, and run bioinformatics pipelines. And in addition, Genome has this automatic scalability and proven cloud computing architecture, which is built in. And that allows you to process these large scale data sets with great efficiency. And that's just what makes it really easy to use and to work together with groups like Puya's at Self-Decode. So, for you out in the audience, whether you're really looking to run pre-made workflows right out of the box to solve your bioinformatics problems, or you're looking to develop new solutions, custom solutions to kind of solve your science and business needs, Genome really has you covered. So we'd love to hear back from members of the audience, to have people reach out. On the right hand of this slide, we've got a QR code, so you can look us up. You can also check us out on the web at almaden.io, or reach out to me personally, and we'd love to show you how the platform can help accelerate your research and work together to show how it can be applied to your organization's problems. So thank you so much for your time, and we'd love to take any questions that come from the audience. I'll turn it over.
Yeah, thank you very much, Mark, for this quick introduction to your product. And also thank you to Puya for your nice overview of your work at Self-Decode. As a quick reminder, if you have a question, please type it in the Q&A panel. And we would also like to ask our attendees to take a brief moment after the webinar has finished to take our exit survey and provide us with your feedback. So let's see what we have for questions. Yeah, here's a question that came in during Puya's presentation when you were talking about the reference-based versus reference-free phasing. And the question is, how is this approach viewed by the FDA? Can you say anything about that?
Like I said, originally this method was really used for research purposes and wasn't really used in any kind of setting where you need FDA approval. Now, that being said, as I understand it, there is actually a couple places where it's used. So I think it's fine from an FDA perspective. Now, obviously, if you were trying to build a test based on imputation, you most likely would have to have we'd have to have a stringent cutoff of where your imputation is actually accurate or not. But from a regulatory standpoint, it should be fine. Currently, there's a lot of AI models being approved by the FDA. and so on. So I think it'll be fine. It's just that you would probably have to use a much more stringent cutoff of where you're imputing correctly and where you're not imputing correctly, if you had to, take this through an FDA regulatory process. But I don't see a particular problem with it. And it's just on us. But I do know a couple other groups that are actually in the process of trying to, get a microarray base filled by an imputation method through an FDA process for polygenic risk score. And I do know what they basically decided is just we're going to do all the common variants. So they didn't see a problem with that from their FDA oversight. So I don't think it's going to be a major issue.
Okay, that's good to know. Great, thank you. Other question for you. Would it be possible to bring pedigree information into the imputation process?
So yeah, but so typically none of these. So there are like some people who use that information for the different ways of doing imputation, but this kind of information is it is possible. Not that many people do it because if you, for instance, like I'll give you an example. If you're, for instance, sometimes adding more information to these statistical things don't actually yield better results. Sometimes what they do is they kind of make it less accurate in other use cases. But yeah, you could theoretically do that. Whether it's going to give you a higher overall result at the end or not depends on kind of the statistical framework you use or not. So, but yeah, possible.
And then a very quick technical question: what would be the smallest number of SNPs and individuals to do imputation on?
Okay, so this, you need, early on they only had 120 reference samples. So, if you have 120, they got some sort of data on there. So, if you have one target and 120 references, you get something. Now, how accurate is going to be that? It's, it's not going to be the world's most accurate thing. It's not really a idea of possibility. It's really an idea of how many is needed for your accuracy that you're going for. Basically, the more variants you have on your genotype chip, the better it is, and the more references you have, the better it is. Now, there is a couple of things you should be aware of. Really old genotype chips that weren't properly spaced out, like the first ones that came out, it's really hard to do imputation. You need to have at least one marker from the haplotype, from the section of DNA you're interested in order to find the missing pieces of information. If you don't have any observable data there, if you don't have any genotype data there from that section, you're not going to be able to impute it. So once again, it's a question of what's your use case. Let's say you have a genotype chip where you only have 50,000 SNPs. Well, you're not going to be able to impute the whole genome based on that, but you will be able to impute larger sections of those 50,000 SNPs. A way to think about imputation is, almost it's always like, okay, I have a SNP here. This allows me to find the information on this larger block of DNA. And the whole idea is, well, if I have enough well-positioned SNPs all throughout your genome, you can find information from all over the chromosome, right? So it's really a use case question.
Right. Okay. What else do we have? Are there cases where phasing can be avoided altogether, and what are those cases?
So actually, some of the software phases it while you impute it, so you don't need to even pre-phase it. But it's impossible to impute without the well, not as impossible, but the whole idea is if you're trying to figure out what chunks it is, you basically need to have them phased in one form or another. But you don't. Sometimes I believe it's Beagle. Don't hold me to this, but I'm pretty sure it's Beagle. You can actually give it just data; it'll phase while it imputes at the same time.
Okay.
So you don't even have to worry about that part. I believe Beagle 5 does that. So there are cases if that's what you mean by avoided, yes, that's what you mean by avoided. But
okay.
Most of these hidden Markov models they're all haplotype imputation models, so you need to have them phased in some sort. But the software can take care of it in many cases.
Yeah, and then this is probably something you are not interested in personally with your business, but what about using this for non-human species? Do you know anything about that?
Absolutely. So a couple of things to be aware of. You can use this for any species. That's, as long as it's eukaryotic. It basically takes advantage of eukaryotic DNA, right? All eukaryotes inherit DNA in chunks, and they all have recombination, stuff like that. The key, though, is you do need a decent amount of reference samples. If you're, for instance, I'll give you an example. I mean, I'd love to do this, but you could definitely do this in dogs, right? But you would need to have sequenced a good amount of breeds because, it's not one breed's not the same. So the amount of reference samples you have, but it's absolutely able to, and in fact, it's used a lot in non-human use cases. A lot of different companies, for various reasons, they want to, like, for instance, genotype array a bunch of, crops. They don't want a whole genome sequencing. So they have some reference panels, and based on that, they can, do that. But yeah, it can be used in anything. It's completely agnostic to the what species it comes to, so long as it's, it undergoes recombination and has linkages to galactose.
Right. Yeah. Mm-hmm. Okay, great. Good to know. And then, I mean, this is all what you talked about was in the context of GWAS and using microarrays, but there's a lot being done with sequencing nowadays. And there's a question about how does long-read sequencing in particular change the problem of phasing and imputation?
Well, that's a great question. I love that. So imputation is also used with sequencing data. Okay, mm-hmm. So this is cool. So, I give an example of the microarray because that's typically what's been used for. But, a current hot thing is what they call, like, low coverage or low read sequencing, or whatever you want to say. It's like, instead of doing a 30x, someone does like a 0.1x or 0.5x, 1x, and then imputes to fill in the rest.
Right.
And then also, but it's a little bit different. I got to tell you, it's a little bit different. So the slight difference is when you observe your genotypes from the microarray machine, you're assuming it's 99% accurate. So your hidden Markov model, these are parameters that go in there. You're assuming it's pretty accurate. Now, if it's a low coverage, it's not the same. So, but the same basic concept works. Just the parameters change. They have their own hidden Markov models. In essence, they're also Lee Stevens hidden Markov models. The same thing works even in that. And I'll give you a use case. I'm not sure anyone has done this, but someone probably does it internally somewhere. I mean, it's perfectly even possible to take these long read sequences. The new machines, they have accuracy issues. Now, they're great at giving you large chunks of DNA, but they have accuracy issues. And then you could probably just set one of these up with the imputation panel, and based on that, improve the results of your long read sequencing. So there's great interplay here. As that new technology comes about, we're going to get better phase data, which is going to make our phasing work better. And additionally, you can use these same algorithms with some slight variations. You typically have their own algorithms for it, but once again, they're very similar, at least in Markov models. So, but yeah, that's a good question.
It seems like it could be useful to kind of check how well the phasing is doing using long read technology as well, since inherently, since the long read technology is only doing one haplotype at a time, you get much more confidence in the connections or the phasing between the variants there, and so it could be also used to check your methodology.
Yeah. Mark, here's a question for you. So the platform that Almaden Genomics offers the possibility to perform imputation in an automated way. That's the question. Does it do that?
Yeah, so for workflows that we have that are pre-built, you can just bring in your data and start to try it out of the box. Now, for something like imputation, there's still going to be parameterization that you're going to check, as well as being able to check it against your specific expectations of the data. And so a workflow can be usable out of the box to give you that start. I still think that you're going to want to be able to modify the parameters of that workflow in order to make sure that it's optimized for your use case. But absolutely, and that's part of what Genome is for, is having those pre-made workflows in addition to being able to build your own.
Okay, and then maybe a related question: how many tools are currently available on Genome, and are you adding new tools?
Yes, that's a great question. We're adding new tools all the time. I wouldn't be able to give you the count as of today because our bioinformaticians are bringing them in constantly. I'd say we have over 100 at this point. And we have massive expandability of the platform. In fact, our users can bring in their own tools using a graphical user interface as well. So bringing in your own tool, whether it's your custom code, a piece of open source software that we haven't brought in ourselves, or bringing in kind of the latest and greatest tools, something like Selfie. If you needed to bring in something that was published a week ago, that's something you can bring in very rapidly on your own using a graphical user interface. It takes about anywhere from 15 minutes to a half hour to bring in any given tool that we don't already have wrapped. So
Okay.
we're expanding our tool collection all the time, but you also have the ability to expand it yourself in a very simple way.
Good to know. Great. Question probably for either of you. Are imputation techniques suitable for single cell genome data? I mean, I guess they are, as long as it's different people, right? But what's your take on that?
Yeah, I don't. Obviously, if I understand the question, they are. So DNA is DNA. So if you have some DNA, but there could be some things here I don't fully understand. If I would love if that person actually sent me an email, tell me exactly why they were interested in this. But yeah. The thing about it is, the little I know about single cell, yeah, I'm actually interested if that person contacts me a little further discussion.
Yeah,
on that.
okay.
Yeah,
Maybe
on the surface, they absolutely suitable, but I am not sure why you would use in single cell data because of the fact that I thought most people are just doing because of the quality was so bad that most people had to do whole genome sequencing on it anyway. So I'm a little.
I think that one place that it might be really valuable is that you're getting differential coverage, Puya, right? So in the same way that you want to unify multiple different GWAS studies or multiple different sequencing studies where they're sequencing different parts of the genome, if your coverage isn't great per cell, finding a way to unify that data set could have tremendous value.
That makes sense. So there you go. Mark answered. I was wondering what they wanted to use. Yeah.
But it's a cool question. I would love to see some exploration there, purely from a more science perspective. But yeah, very good question.
Yeah, thank you. Thanks. Question for Puya. Is selfie available for academic use?
Absolutely, it is. We have not. It's currently undergoing final touches, so we can submit it as a paper. So you can't find it online, but it's absolutely available for academic use for anybody who's interested. Just contact me. I believe my email is going out on this thing or not. But anyone interested for academic use or even a collaboration on it for academic use, please contact me. We would love to collaborate. We could even help you set it up and run it and all that stuff. So please contact me.
Yeah, okay. And a question that just come in, can you comment on the concordance rate between whole genome sequencing and imputed data in general, I guess? And what is the advantage of using whole genome sequencer compared to imputed data? Do you even have to do this? And is it is the advantage only on the discovery of rare variants?
I don't actually know what the concordance rate is between the two off the top of my head. But I will tell you this, the real advantage of doing whole genome sequencing is, the rare variants. That literally is. It's literally by far the most powerful aspect of it. When you do whole genome sequencing, especially if it's like 30x, when you say this person has a really, good loss of function rare variant knockout, like you figured out the protein is knocked out here because of this rare variant that you've just discovered in 3 people. They have it. Imputation is as good as, like, for instance, changes are to imputation. It's just never going to be as accurate. There's always going to be some uncertainty whether they have it or not. So that's really the real advantage of whole genome sequencing. But it comes at like a huge, massive costs, right? Not only the cost of doing the sequencing, but the cost of storing the data, processing it, and so on. And so when people have hundreds of thousands of samples, it just makes more sense to do imputation. But really, the real advantage is that you are more certain, and this really matters when it comes to the rare variants. Additionally, whole genome sequencing, depending on the kind of techniques you use, it might be more possible that there's certain kinds of variants you might be able to figure out more, like structural variations. That's just not going to happen with imputation. Those kinds of things, especially if you're doing like long read sequencing and stuff. So that's really the big advantage.
Okay, great. And Mark, can you quickly comment on academic use of genome? Is it a commercial product only, or is there any other academic usability?
Yeah, we're currently working with academics already, and so I highly encourage you to reach out and connect with us, because we'd love to discuss how we can bring it to your lab or to your organization. Absolutely.
Sounds good. Let me see. A question that just came in How complex would it be to impute a low-pass genome with 2x mean coverage? Very specific question. And wouldn't the phasing be difficult for that?
So, low pass, it's actually, it's very accurate. Some of these newer statistical models they have, they're doing like not even 2x. It's like literally 0.5x, 0.1x. They've got it working well. So here's the thing is that phasing in theory, you would think would be difficult, but what happens is when you have like bigger chunks of the DNA and it's like falls on top of each other, the phasing algorithms get a little bit better actually. So, like I said, they're Lee Stevenson Markov models, but they don't use the same thing. Like you don't use Beagle for that. You don't use Shape It for that and so on. They all work similarly, right? They have the same general idea, but there's specific ones that are designed for low, for low pass sequencing, for these kinds of low coverage, whatever you want to call it, sequencing, like 1x, 0.1x. They work on the same general idea, and they give very good results. They give very, very good results. It's quite startling how good the results are. So,
right. Okay. Let me just see. So there's something earlier in your presentation, Puya. You showed there was an increase in accuracy between the reference-free versus the reference-based results. Could that have anything to do with differences in population?
Yes and no. So it really kind of depends. Obviously, I'll give you an example. If you're testing your phasing with a population you don't have samples of in your reference, it's going to get lower results. So in that way, the question is right. Yeah, yes, that could affect it. In our case, that was not the case. We kind of control for that, so we know that's not the case. But yes, you could get, if you have your targets are a different ancestral background than your reference, or vice versa, or any way you want to work it, their accuracy of phasing is also going to be a little bit worse if you're using reference-based. Now, if you're using reference-free, you're doing all these together, it kind of gets around some of this stuff.
Yeah, okay. And a related question related to the accuracy of imputation in different populations, what could be done to improve that? Would more data help or an improved
Absolutely.
algorithm?
The answer to all this is always the same: more data. All right, so that's always the answer in genetics. That's thank God the NIH believes in that. So you keep saying more data, more data, more data. So the answer is always more data. You really do. Ideally, you would want tons of references from tons of different ancestral backgrounds, right? Tons of different kinds of people that are admixed, that have these kinds of ancestral backgrounds. This is always going to improve accuracy. It's a statistical model, and it's a great statistical model, but it's not like a magic show. Like it can't figure out what haplotype someone has if it's never seen that before in reference, right?
Right.
So at the end of the day, you have to have the data in order for it to learn. It's not a magic show. So the key is always to get better and deeper and reference sets that are not just like we've sequenced 1 million British people living in England. So.
Okay. Oh yeah. One more question that just came in. What is the influence of having a database of a mixed population in the prediction of a 1x genome?
I don't fully follow the second part of the question.
Yeah, I don't.
But I think I might know what it. And since we got 2 minutes, I'll just answer it. This is the last. This might be it.
Yeah. Right.
It's the same idea. And so I think what they're saying is that if you're doing this kind of low low coverage ones, is it the same? It's always the same. You need different ancestral backgrounds in your population in order for accuracy to be improved. The reason is it's purely biological. It has nothing to do with the algorithms, right? Different populations of ancestry, what really sets them apart is the kind of like linkage blocks they have, how big they are, how much diversity you find in. You can't get around this. The only way to get around that is if you have those in a reference and that you could see it, your algorithm could see it, infer it, learn from that, and use that to do things properly. But you can't get around it. If you have some ancestral populations, because they're newer, they've had greater population bottleneck, they're just less diverse. And you're like, oh, wow, imputation works great in them. But other ancestral populations, like a lot of these African tribes that have been over there, they have a lot of genetic diversity, right? So you need way more references in order to impute those with accuracy. You can't get around that. You have to have a diverse panel in order to impute correctly.
Yeah, okay, that's a good closing statement. And this is also all the time we have for questions today. So let me thank our speakers, Pooya Yazdi and Mark Kunitomi, and our sponsor, Almaden Genomics. And as a reminder, please look out for the survey after you log out to provide us with your feedback. And if you missed any part of this webinar or you would like to listen to it again, we will send you a link to an archived version via email. So with that, thank you very much for attending this genome webinar.
Thanks, everyone.